We're now on Chapter 3, which is all about the building blocks of life. It's called the molecules of life. And much of our studies here centers around what are called macromolecules. Macromolecules, the prefix macro, just simply means big. And our bodies and living organisms consist of about 4 major macromolecules: carbohydrates, lipids, proteins, and nucleic acids, and we'll talk about each one in particular. One thing many of these have in common is that it takes smaller molecules to build them. A large molecule made out of repeating units is called a polymer. The prefix poly simply means many. The different units that build the polymer are called monomers. So for each of these, one of the green circles is one monomer, but when you put it all together, you have what's called a polymer. So think of monomers like building blocks or links in a chain. We learned in last chapter that bonds form between molecules. These lines between the monomers here symbolize those bonds. In particular, these are covalent bonds, which are the strongest of bonds. Remember, that's the sharing. The sharing of electrons when that happens. Alright, in this case here, this is a little background that if you learn this here, this will apply to all of the polymers, aka the macromolecules that build living things: carbohydrates, proteins, lipids, and nucleic acids. So some of the the simple rules that go behind building and tearing these things down. Alright, so if we think of each of these green circles here as a unit that builds a polymer, we need to put them together. So we need to create a covalent bond right in here where you can see my cursor circling. Alright, to put them together, well, we're going to have to get rid of some of the atoms that are in between here in order to establish this bond with that one. So in this case, this H over on this monomer will combine with this OH on this monomer to form a very common, familiar molecule called water. And then the bond is established. Alright, so because we took water out to synthesize this polymer or macromolecule down here, it's called a dehydration synthesis. Dehydrating it, taking water out, and then synthesizing the molecule. It's like one monomer donates the OH, the other the H, and voila, we get water. Again, that's a dehydration synthesis. It also goes by another name called the condensation reaction. This, of course, is going to require energy and enzymes to help this happen and to help it happen efficiently and quickly. So I like this picture here of this yellow thing coming in here symbolizing the enzyme. And enzymes typically work by holding molecules in place so that the right areas can get their bonding done. That way this reaction will happen much more faster than if the enzyme had not been present. Now the opposite, something that your body has to do very, very commonly, in fact every time you eat it's doing this. We have to take apart those big polymers, or aka macromolecules. When you eat, you consume things like carbohydrates, lipids, and proteins. You have to break them down into their parts, their monomers, so that they actually become useful to your body. And then your body takes them and rebuilds them into the things that make you. So in the digestion or in the breaking down of the polymer, we have the opposite happening. So instead of a dehydration synthesis, we have to take the water and put it back. So that one of the H's will go on a molecule here, one of the OH's, and here we have it. When it's all said and done, our monomers are now separated. We've digested it. This is also called a hydrolysis, and this can happen for each of these green circles that you see here. Like all the reactions in our body, it requires enzymes. Here comes our purple enzyme along the way to help things out. Again, enzymes work commonly by holding molecules in place, allowing bonds to be formed or broken much more easily than if the enzyme was not present. Sometimes enzymes even put pressure on the molecules themselves to get them to do their reaction and form and break their bonds. These types of reactions, digestions, and dehydration syntheses are happening all the time in your body. So let's talk about carbohydrates. In the pictures here we have some very common and yummy carbohydrates. When you think of carbohydrates, you probably think sweet, because, well, carbohydrates are made of sugars, and, well, sugars are sweet. Carbohydrates' main function in our body is for energy. Here we have a monomer, or think of it as one building block of a big carbohydrate. This is a sugar right up here in the upper right corner. It's the common sugar glucose. Carbohydrates get their name because they're made out of carbons, hydrogens, and oxygens, and lots of them. The interesting thing is that the hydrogens are always in a 2-to-1 ratio with their oxygens, just like water. So that's why we call them carbohydrates. Basically, they're hydrates of water, or hydrates of carbon. See how this molecule here, this is glucose, how the hydrogen to oxygen ratio is 2-to-1. Again, think energy and energy storage and some structure as well in your body as the functions that carbohydrates serve. They really are what you call polymers, long chains of sugars. And that's what we get when we have a carbohydrate. The monomer is the sugar, the polymer is the carbohydrate. Starches and cellulose are carbohydrates. Sugars tend to end in OSE, such as the word glucose. You've probably heard of fructose and sucrose. All of those have in common that they have an OSE ending. Down here again we have glucose. It's a 6-carbon sugar. We can see one carbon right here, but also think that at each of these corners there's also a carbon that is just unsaid, except for right here where we have an oxygen. Ribose, a common sugar in RNA, is actually a 5-carbon sugar. And here we have a 3-carbon sugar. That one does not form a ring or a hexagon or pentagon. It's just pretty much a straight line. There's only 3 carbons in it. You'll probably notice that macromolecules that make up our body have a lot of carbon in them. What we're studying here is a section of chemistry called either carbon chemistry or organic chemistry. Because of the way carbons want to have 4 bonds on each side, like we discussed last chapter, they can form these long chains. This one again, this glyceraldehyde here, only has 3 carbons, but these chains can actually get pretty long. Some of the functional groups here, we commonly see a C with an O. This is called a carbonyl group in some of our sugars. When it's on the end like this, it's called an aldehyde. And when it's in the molecule like these here, it's called a ketone. Again, these are all sugars here. There's our common one called glucose right here, a 6-carbon sugar. On the previous slide, it was in its regular kind of hexagon ring shape. Here it is nice and spread out in a chain. We could look at it either way. In a solution, though, it would most likely form a ring like this. Carbons are numbered. So this is our first carbon right there. So we call that the 1' carbon, the 2', the 3', the 4', the 5', and the 6th one hanging up over here on the top left corner of this glucose molecule here. Here we've left out the H's and some of the O's. We're just pretty much focusing on what it is on these corners here, especially the carbons. This becomes important when we study other molecules like DNA, who also have this ring structure of sugars. It kind of helps us orient what side of DNA it is we're working with. Such as, are we on the 3', 5' side, or are we going the other direction, 5' to 3'? One thing also to remember is that there's energy stored in all these bonds here. Remember, when energy is transferred to something else, we can get things like movement. Monosaccharides. Let's talk more about sugars. Monosaccharides are just a 1 monomer sugar. Glucose is a good example. Disaccharides, the prefix di means 2, are 2 monomers or 2 sugars joined together. Common table sugar like sucrose is an example of that. Polysaccharides, poly means many. Well, these are large polymers, so they're going to be large or long chains of these things strung together. Starch and cellulose are a great example of that. All right, let's put our knowledge to use now. We already talked about what a dehydration synthesis was. All right, so we have our 2 glucoses here, and we want to join them together. So we're going to take the OH on one of them and the H on the other, and we're going to form water. That's our dehydration part. And then we'll form our bond and join them together. So what we just formed here, when we put 2 sugars together, it's called a disaccharide. Maltose is an example of a disaccharide. Maltose is commonly used in beverages like the one pictured here. Here's another example. When you take a glucose and you combine it with a fructose via a dehydration synthesis, you get common table sugar, sucrose. And as we know, that can be pretty yummy. Polysaccharides. It's actually pretty energy efficient to build these things. It doesn't take a lot. Therefore, they're very good for energy storage. In plants, it's stored as starch. So think of this, when plants do photosynthesis, they make their own sugar, but they make it in the form of glucose. And when you string glucoses together to store it for later use to eat, well, that's starch. Plants need to eat too, so we kind of always just focus on the photosynthesis part of it, but plants really do need to eat, they need energy. So after they make their sugar glucose, they store it as starch. That's a lot of dehydration synthesis going on there to combine all those chains of glucose to make starch. But again, it's little energy to do that, so it's easy for plants and animals to make this polymer. In animals, we call it glycogen, and we tend to store it in our liver and our muscles. Again, both of these have in common that they're chains of glucose. Well, there's a couple other types of carbohydrates here. These are really big ones. Cellulose is a carbohydrate, and chitin is a carbohydrate. Again, just like these down here, they are chains of glucose, just maybe strung together in a different way, in a different bonding pattern. But the problem is we don't have enzymes to break these down, to do a hydrolysis or a digestion on them. So when we eat plants and we're getting a lot of cellulose, this is also called fiber, like eating lettuce and the shells of corn. We can't digest that, so it helps go through the digestive tract, and it pulls water in, and it basically keeps us regular. Chitin, same thing, can't digest it. It's got a little bit different bond on it. Chitin forms the exoskeleton of bugs, insects, crustaceans, and it's another reason why when we eat seafood like this, we don't actually eat the exoskeleton. Granted, it is a carbohydrate, it's a polysaccharide, but we can't digest it. We don't have the enzyme to do it. Here's a couple slides to show us some pictures of the difference between what starch looks like and what glycogen looks like. Having in common, these little hexagons here are chains of glucose, but a little bit different shape to each one. And that's due to the bonding. Both are for energy storage, but one's for plants, one's for animals. Because of the different way these things bond, that's basically why you may or may not have an enzyme to break it down. When bonding occurs in different places, then it changes the structure, and therefore enzymes are very, very particular about the structure of the molecule that they work on. And if it's not structured just right, if things are not in the right place, like this alpha glucose has the OH down here, the beta one has the OH up there, an enzyme may not work. Things like cellulose in our body. We can digest this one if the OH is down there, but not this one because the OH is up there. Structure determines function. Here we go. Enzyme works. Good to go. And the cellulose does not work. Nope. Alright, but look at the shape of how this is in the middle here. Again, cellulose in plants, and not going to be something that you can digest. There are some animals that can do it though, but it sure does take a lot. It's too bad, because cellulose is one of the most abundant organic compounds on Earth. Again, an organic compound is something that contains carbon. Herbivores have evolved quite the system in order to digest it. It involves having a lot of bacteria and many chambers to their digestive system, and even having to regurgitate and re-chew. That's how tough cellulose is to digest. A little help from our bacterial friends. If we were to examine the gorilla feces here, they're not as good at digesting cellulose, so we would find a lot of roughage, I guess, in their feces left over. Here's a picture of a cow digestive system. Look how intricate that is, with arrows coming back up for that regurgitation and re-chewing process and re-swallowing. They're also called ruminants. And even then, there's still a lot of nutrients left in their feces, so that organisms like these insects here can feed on their feces and still get a lot of nutrients out of there. Plus, since the cow has already put in some work to digesting it, it makes it easier for these little guys here to digest it. Alright. There's a young lady here who's going to get some good carbohydrates there. I wonder who that is. Now, let's talk one of my personal favorite macromolecules that build the body, and they are, well, fats and oils, also called lipids. You have to admit, they taste pretty good. Peanut butter, mayonnaise, all these good things here. When we think lipids, let's think long-term energy storage. There's some similar things about them to carbohydrates, but yet some differences. It's very concentrated energy as well. Alright, just like carbohydrates, they're formed from carbons, hydrogens, and oxygens. However, where the carbohydrates had the 2-to-1 ratio of hydrogen to oxygen, these don't. For these, we tend to see again those long hydrocarbon chains, long chains of carbon and hydrogen. And again, that's why this is part of that group of chemistry called organic chemistry or carbon chemistry. There's some different kinds of fats. There's fats, phospholipids, and steroids. One difference from carbohydrates is that these things do not form polymers, so that means that they're not made out of repeating units or repeating monomers, but tend to have this as their basic structure, as you see right here. Alright, so what makes up a fat? Well, we have this area right here. It's a 3-carbon molecule called glycerol. On the side where we have OHs, this is where we can join the other part called a fatty acid chain. Alright, just like with the carbohydrates, to join our fatty acid chain with our glycerol, we've got to do a dehydration synthesis, meaning we have to take a hydrogen away from this molecule and an OH away from this fatty acid, and in doing so, then we will join these together. Remember, that's called a dehydration synthesis. Oh, got to have an enzyme to help that out. And if we do that, I'll back up here a little bit. There's actually, if you take a look here, let me get rid of that enzyme out of the way. There's actually 3 spots here where we can add a fatty acid chain to it. So if we use all 3 spots, our completed fat will look like this. 1 glycerol on the end, this is still a glycerol, a 3-carbon molecule, and then our 3 fatty acid chains hooked on. So that would mean that we would have to do 3 dehydration syntheses to make this whole molecule. When we join an OH with what's called a COO group, COOH group here, that's called an ester linkage. In a carbohydrate, it was called a glycosidic linkage. So there it is again. Kind of makes like a fork structure here. Again, 3 dehydration syntheses to connect all 3 fatty acid chains. Therefore, we'd actually have 3 molecules of water when we were done with this. We would need 3 enzymes. We learned what these words meant last chapter. Water, in last chapter, we talked about it being a very polar molecule, being that there was unequal sharing of electrons with the oxygen to the hydrogens. It leaves a bit of a charge on the molecule. Well, that's water. It turns out fat is nonpolar. There's really no differences in charge among any certain place here in the molecule. It's all pretty equal. That makes it the opposite of water. This is a reason why fats do not mix well with water. They're called hydrophobic, water-hating. That's why if you're going to wash something that's fatty, you're going to have to add some detergent. Again, all these carbons and hydrogens, all the energy stored in these bonds, this is very concentrated energy here. Well, we know what can happen when we eat too much fat. It can get stored. All that concentrated energy stored up can puff up those fat cells and cushion organs and really insulate your body. Think whale blubber. Why do you think we like fatty foods? That's a good question from the penguin up here. Well, I guess that's evolution's way of kind of like protecting us. The fats taste good, and it's like a reward that we eat them to create that fat to insulate our organs and such. It's like a reward when we eat it. In times of famine, yes, that's good, but it turns out many of us today are not in a famine situation. So really, we don't need to be eating that much fat. If it's a saturated fat, you often probably hear about saturated versus non-saturated fats. Well, here's the difference. In a saturated fat, every single carbon has a hydrogen bond here on these 3 fatty acid chains, and it makes the molecule, it's really regular here. All of these fatty acid chains sit nice and flat on top of each other. Therefore, you're able to really scrunch a lot of these fat molecules together. And so these saturated fats tend to be solid at room temperature, like butter and lard. And, well, being that they're so able to clump together so well, they can do that inside of our arteries and cause atherosclerosis, often called hardening of the arteries because of plaque deposits. So you have to watch out for saturated fats and not eat too many of those because they like to clump together. Let's look at the other one. Unsaturated fats. Well, some of the carbons tend to not have a hydrogen in certain places, so they end up double bonding to their neighbor carbon, and it creates these little kinks in these fatty acids. And so taking a look here, you can see how the fatty acid chains don't sit flat on each other, and so you can't really scrunch them together very well. So these tend to stay liquid at room temperature. These are mostly from plants and fish fats, whereas the saturated fats are more animal fats. And there's a difference in the look. Again, a little reminder there with the big red circle. Those are not as healthy as the unsaturated. Phospholipids are another type of fat that maybe we don't hear about as much. Phospholipids make up our cell membranes. We'll be talking cell membranes in chapter 2. And if we take a look at our phospholipid here, we've got a couple different models. I like the one on the far right. This area here, that's the fatty acid chains that we saw. In a phospholipid, there's only 2 fatty acid chains. One of them has a kink to it. And up here is our glycerol. But there's this phosphate group right in the middle. And this tends to give this area of the molecule a negative charge. So this is an example, unlike regular fats, this is a polar molecule. It's got a charged end. But the other end is uncharged, so this end is still hydrophobic. It doesn't like water. This end, because it's charged, it doesn't mind water. So when we put these in water, they tend to form rings or membranes just naturally. This becomes very important for setting up the structure for our membranes. Here's an example of that. The circle part, that's the top part of that phospholipid, and it's the hydrophobic part because it's got that charged phosphate group. And these ends down here in the middle, excuse me, I think I said the wrong word there. This is the hydrophilic end. These ends down here are hydrophobic. They're the non-charged ends. And so when you put them into water, these hydrophobic ends keep trying to get away from the water that they're in, and then they form these circles like this. And it's kind of like a little membrane. And if they really want to get away from water, they'll do this over here. They'll form a bilayer. So these little ends touching each other, they're away from water. Out here in the blue, here's the water. But these are the charged hydrophilic ends. They don't care. So our cell membranes actually have a double layer just like this. It's called a phospholipid bilayer. And, well, can help hold our cells together. Kind of important. Soap can help break these membranes down. That's why we need to use soap to help us wash away the fats. Steroids are another example of fats or lipids. But what's different? They tend to form rings. Cholesterol is our most basic steroid molecule. And sex hormones like estrogen and testosterone. Cholesterol is important. We kind of give it a bad rap, but it is actually an important part of cell membranes embedded in the phospholipids. It's kind of your body's building block to creating other steroid molecules in your body like testosterone and estrogen. So really kind of an important molecule, I would think. Problem is we eat too much of it. Our liver can make quite a bit of it from just eating a little, or excuse me, quite a lot of it from just eating a little bit of cholesterol. So we really don't need to eat a lot of this. And when we eat a lot, well, this happens. Our cholesterol can build up in our arteries. Here's some examples of some steroid fats. They look very similar, but just adding little functional groups like an OH here and a OH there or a CO over here just makes all the difference. Looking at testosterone and estrogen, there's not a lot of difference between those two molecules, but look at the outside of the changes, the differences between male and female that just those little molecule groups make. Build some lipids. Next, our next macromolecule are proteins, another one of my favorites. Look how delicious those are. Think of proteins as your multi-purpose molecules. They build a lot of stuff and have a lot of functions in our body. I also like to think of them as the workhorses of our body, but man, do they have complex structure. It's a very diverse group. Here's some examples. Proteins can be our enzymes like the pepsin that helps digest food, the enzyme DNA polymerase which builds our DNA, the proteins in our skin like keratin and collagen that are the structure. They function as carriers and transporters, such as the hemoglobin on our red blood cells carrying oxygen, and aquaporin on cell membranes transporting water. They act in signaling mechanisms in our body as hormones, as receptors on cell membranes to receive molecules. They act as defense. Our antibodies that we make are actually proteins. And they act for movement, like the actin and myosin in our muscles. And for storage. Lots and lots of different jobs. Just like carbohydrates, proteins are made from monomers. Therefore, to join two of them, we must do a dehydration synthesis, or to tear them apart, digestion or hydrolysis. There's many different amino acids. Amino acids are the monomer that build proteins. Though there's many amino acids, only about 20 are found in our body. So all of the different proteins that have so many different functions in our body are built from only about 20 different building blocks. But considering that when you put them together in so many different ways to make a polymer or to make the protein itself, that there's so many different ways to do that. There can be so many different proteins. The word polypeptide here is kind of a word for a smaller protein or an unfinished protein. Some different protein pictures here. An amino acid looks like this. It's got a little nitrogen group here on the side. That's called an amino group. And over here, a carbonyl group. When you have a C with an O, that's called a carbonyl group. And again, we have to take an OH from one and an H from another, do a dehydration synthesis, and then we can start building our protein. And if you do this over and over again to the building blocks, you'll eventually have your polymer or your protein. But then you're not even done yet. When you string the chain together, then you have to fold it. Like you see, hemoglobin here has lots of folds to its chain. We have to get the right structure or the protein won't work. Alright, so a little bit more close-up view of amino acids. There's our amino group, our carboxyl group. And then if I go back here, all amino acids have this in common. But the R group is what's different about them. And there's 20 different amino acids, so 20 different R groups. Think of it like our alphabet. We have 26 letters to our alphabet to make an unlimited amount of words. Well, you can make an unlimited amount of proteins from the 20 different building blocks called amino acids. I'm going to skip through these slides here. This is getting pretty specific for what we need to know. But amino acids can come in nonpolar form. They can come in polar form or charged. Remember, water is a polar molecule. Some of them can donate hydrogens, being they're acidic, and some can accept. Some contain sulfur, reason why eggs smell like sulfur when they're rotting. So this also is important on how proteins fold, because sometimes when they fold, a sulfur will connect with a hydrogen. And you get two of those together, the two sulfurs connected, and that's called a disulfide bridge. Must be a lot of those in perms. That's why those smell like rotten eggs. The actual bond is called a peptide bond that forms them. Remember, in carbohydrates it was called a glycosidic linkage. In fats it was called ester linkage, and here it's called a peptide bond. But they have in common, we still have to do that dehydration synthesis. Make a molecule of water. They have a direction. Here we've got in the purple squares, each of these is an amino acid joined with a peptide bond via the dehydration synthesis. On this end here, or N, yeah, the end here, we've got the amino group, so that's the N terminus, nitrogen down there. Over here, the C terminus for this carboxyl group. So that means when we make proteins, they only grow in one direction. Now let's talk their 3D structure. Our proteins are built in our cells at a little place called the ribosomes. And to do that, we have to put together the amino acids in the right order. We'll talk later about how DNA is responsible for your body knowing what order you have to string these amino acids together in to get the right protein so that it functions correctly. All of these amino acids here have kind of a nickname. And look at all of these. This is a pretty simple protein here. It's called lysozyme. It's kind of an antibiotic protein / enzyme that's in your tears and mucus and helps to kill bacteria. This one's pretty short. It's only about 129 amino acids long. Most proteins, though, are thousands of these things long. But get the right order or it won't work. If I, for instance, took this little LEU out here, this protein might not then be shaped correctly and it might not work correctly. Again, DNA is responsible for telling your cells what order to put these in. This is called the primary or 1-degree structure. A great example of a problem with this primary structure is in the protein hemoglobin. Most of us have this regular order here of amino acids, but there's some people who have a mutation in their DNA And therefore your cells put together this order, a val instead of a glue. Again, those are nicknames for amino acids. And because of just that one little change of amino acid, it changes it to a hydrophobic amino acid, and well, then it causes that protein hemoglobin not to work. It's a mutated form, and it causes red blood cells to actually sickle and shape, kind of turn goofy. And remember, red blood cells, hemoglobin is in charge of carrying oxygen, and when cells are shaped like this, it doesn't work. So people that have this disease called sickle cell anemia due to this one mutation here get very sick. The second degree structure is just kind of a small amount of folding in the amino acid chain. Small amount of folding. Sometimes it's kind of a twisted folding. Sometimes it's kind of like this is a beta sheet here, or beta pleated sheet, but not a huge amount of folding. That's the second degree structure. Again, that's going to be because of the interaction of the amino acids in the chain. The third degree structure is when you do this whole molecule folding here. It's like this whole amino acid chain has folded over on itself. Again, that's because of the interaction of the different amino acids within, and whether or not it's in water, and etc. Different types of bonds. And quaternary structure, sometimes it takes more than one amino acid chain to make these things. They're so complex. Like hemoglobin here has 4 different chains to it. Collagen here has 3. So we put together these unfinished chains called polypeptide chains. And then finally, if it's shaped just right, our protein will work. This is hemoglobin here, if it's done right. This one's collagen. Here's a summary. When our proteins come in contact with excess temperature, such as it's too hot, too acidic, too salty, that can actually break bonds of our amino acid chain folding, and can do what's called denaturing the protein. Once you denature a protein and it's not shaped like it once was, it won't work anymore. And you can't reverse this either. That's why it's so important for our body to stay at a temperature of 98.6, because if we get too hot, our proteins denature. We break those important covalent and ionic bonds holding the different folds together. Here's a little kind of what's to come when we get into our next cell chapters. But this is a model of a cell. In the nucleus, there's our DNA. DNA has a code that's copied by a molecule called RNA. This is the endoplasmic reticulum. It's kind of like little tubes within the cytoplasm of a cell, and also houses these little circular structures here called ribosomes. That's where proteins are made. So mRNA here made a copy of DNA's code. Because RNA is only single-stranded, it can get outside the nucleus and will travel to one of these ribosomes. The ribosome locks on that mRNA strand, reads it, reads its code, reads its order, and then that order tells what amino acid to string together next so that we get the proper chain. So again, remember, this is the 1 degree or primary structure of a protein. Our unfinished protein, which we should probably call a polypeptide instead, can then be kind of vesicled off and sent for finishing at the Golgi apparatus, another tubular structure in the cell. Go through some little changes, bonds will form, and we'll get the right folding. We'll bubble it off again, and then maybe we will ship it via the bloodstream to somewhere where it's needed. Maybe we might even keep it within the cell to use it there. But this is basically how we create and then ship off a protein in our body. Again, we'll get into this more in Chapter 3. How about building or eating some proteins here? Our last macromolecule is nucleic acids, and these are what build our DNA and our RNA in our body, in our cells. So think of them as information storage. And they're going to have some things in common with some of our other macromolecules, especially in the way that they're built and broken apart. Nucleic acids, again, build our DNA. Our DNA stores the information to be able to make proteins, as we saw previously, and can help transfer our information down to the next generation. DNA built, broken down, looks like this. Get into that a little bit. RNA is a single-stranded nucleic acid, said to have a single helix. DNA is double-stranded, so said to have a double helix. They are both built from monomers, just like proteins and carbohydrates are. The monomers that build them are called nucleotides. So each nucleotide has 3 parts: a nitrogen base, a sugar, which is a 5-carbon sugar, and a phosphate group. The sugar in DNA is called deoxyribose, and the sugar in RNA is called ribose. Again, there's our OSE endings, and here's a picture of what each of those sugars look like up here. Remember glucose? It had a hexagon. Well, these two, since they're a 5-carbon sugar, have a pentagon. And it's been found that there are 4 different kinds of nucleotides, but can be categorized into 2 groups: purines and pyrimidines. We usually don't worry about those words. Instead, we worry about their specific names. The 2 purines are called adenine and guanine, and to make it even shorter yet, A and G. And the 2 pyrimidines are called cytosine and thymine. Here's adenine, here's guanine, cytosine and thymine, and then in RNA there's one called uracil. It's called a phosphodiester bond in between each of these monomers. Keep in mind we have to have a dehydration synthesis happening to build these, so we have to take water out, and we can only grow these in one direction, just like in proteins. Here's an even better view of what that looks like. We found that between the A and the T, adenine and thymine, we like 2 bonds, so they always bond together. Between the G and the C, guanine and cytosine, we like 3 bonds, so those 2 always bond together. A's with T's and C's with G's. So that makes this molecule very predictive. We can predict things. If we know one side we then know the other. If there's a G on one side, we know there'll always be a C on the other. So taking a look at this, remember that each base here, like for instance thymine, has a part of it called a sugar and a phosphate group. This whole thing here makes a nucleotide. Here's another nucleotide, a guanine nucleotide, and a cytosine, and then an adenine nucleotide. But when we really talk DNA, we really focus on this middle part here, the A's with T's and the C's with G's. Here's another view of what that might look like. When DNA copies itself, it can break apart the hydrogen bonds in the middle, and then enzymes come in and fill in the other side. That makes replication or copying much more efficient and easy for DNA. Because if there's a G here, well, then it knows to put in an A on the other side. It's like matching halves. Cells have to copy DNA when they want to reproduce. That would be during mitosis, and when they want to make sperm and egg, during meiosis. We'll talk more about DNA in our future chapters on genetics, but we still credit these two gentlemen here, Watson and Crick, with figuring out the structure of DNA by using a lot of research from other scientists. Made a little controversy when this guy in England gave this young lady's work to these two. So we'll talk about that controversy a little later. So this is a X-ray diffraction picture, and if you put a little mathematic equation to this, you can figure out that DNA is a double helix. And Watson and Crick won the Nobel Prize in the 1950s due to that. Unfortunately, working with so much X-rays, Rosalind Franklin here died of cancer. In a molecule of DNA, we should always find that there should be about the same number of A's as T's in DNA, and there should be about the same number of G's and C's in DNA. That's because of their matching patterns. ATP, adenosine triphosphate, an energy molecule in our body, is also a nucleic acid, though not made as a polymer. So it's just like a single monomer that exists to help us move and do things within our body. But just one of these here has a similar form as a nucleotide of DNA or RNA. It's just got a couple extra phosphate groups over here. There's our 3 phosphate groups there. There's a double helix. Alright. If you go back to our book, the opening picture has a picture of some French fries frying. We all love those. It talks about trans fats and how the process of hydrogenating oils creates these trans fats, which have kind of a weird kink to them, and therefore our body can't process them very well. It's kind of an interesting reading. Make sure you check that out on the beginning of chapter 3 there. And then this concludes our notes for chapter 3. Again, I do recommend doing the after chapter questions to help you kind of put it all together and make sure you're understanding this stuff, because it is kind of a big chapter. putting all this chemistry information together.